complex diseases, the ones that they defy simple answers no matter how much research we throw at them. They're a huge challenge because our traditional drug discovery models, well, they just don't capture the full story.
Exactly. They often fail because they treat disease like a series of static snapshots.
Right. It ignores this incredibly dynamic movie that's playing out inside our bodies at the cellular level. We need tools that can be, like cellular historians mapping out how a disease progresses and then turn around and be molecular fortune tellers.
And predict what will actually work.
Yeah.
That whole underlying story, that narrative of how cells change over time, that's everything. If you don't get how a healthy cell becomes a diseased cell, you can't really figure out how to push it back.
So that's what we're doing today.
It is. We are taking a deep dive into an advanced computational tool designed to solve this exact problem. It's called UNAGI.
UNAGI. I like the name. It sounds powerful.
Yeah.
So for our listener who might be trying to get up to speed on this, what's the core mission of UNAGI?
The mission is basically this. UNAGI is a deep generative neural network, and it's built to chew on time series single-cell transcriptomic data.
Right.
It's really designed to bridge that critical gap between understanding what's happening at a molecular level and then speeding up targeted in silico drug discovery.
So it's like building a digital roadmap of the disease.
That's a great way to put it. It lets us rewind, pause, and maybe even reroute the whole biological process one molecule at a time.
Okay, let's unpack this. Where does an architecture like this even begin? We're talking about single-cell data, which is it's messy, it's massive.
It starts with its generative foundation. It's a blend of two pretty powerful AI techniques, a variational autoencoder, or VAE, and a generative adversarial network, a GAN.
A VAE-GAN.
A VAE-GAN. The VAE part, its job is to compress that massive amount of gene expression data, we're talking thousands of genes per cell, into a much smaller, manageable representation. We call it the Z latent space.
So it's capturing the sort of digital essence of each cell.
It's a digital essence, yes.
Okay, that's the compression.
Yeah.
So what's the GAN doing? Is it just checking the VAE's work?
That's a perfect analogy. The GAN is quality control. Its whole job is to make sure that when the VAE tries to reconstruct data from that compressed Z space, the synthetic data it generates looks exactly like the real thing.
So it guarantees the cell's digital twin is actually accurate.
It's high quality and it accurately reflects the original biology. Yeah.
So it's learning the compressed language of the cell perfectly.
Yeah.
Okay, let's talk about the noise. Single cell data is, well, it's famous for dropouts. It's like trying to hear a quiet conversation in a really crowded room. Some genes were there, but you just didn't catch them. What happens if you use a standard model for that kind of data?
Well, if you assume a standard distribution, your model basically pretends the crowded room doesn't exist. It treats all those missing data points, the zeros, as if the gene wasn't expressed at all.
When it might have just been missed.
Right. UAGI is engineered to handle this by using what's called a zero-inflated log-normal distribution, or ZILM. And this is a really crucial detail. ZILN models two separate things. First, the probability that a gene was just missed, that's the zero inflation. And second, the actual level of expression, which is the log-normal part. It just fits the statistical reality of single-cell data so much better.
That's really clever. It's adapting its own math to the limits of the biological measurement.
Yeah.
But there's another layer on top of the VA, right? The graph convolution layer, or GCN. Yeah. What problem is that one solving?
The GCN tackles the noise and sparsity again, but from a different angle context. We kind of call it the cellular neighborhood watch.
Okay.
It builds a connectivity matrix using an algorithm like k-nearest neighbors to figure out which cells are biological neighbors.
I see.
And by factoring in what its neighbors are doing, the GCN layer makes the cell's representation in that Z latent space much more accurate. So if one cell is noisy, its buddies who are biologically similar can help, confirm its identity.
And fill in the blanks from that dropout noise.
Exactly.
Got it. So phase one is all about creating the sharpest possible digital snapshot of each cell. But that's still static. Phase two is where we get to the dynamics, the progression. How does UNAGI start mapping that journey over time?
So once we have these really refined Z's embeddings, we cluster the cells and visualize them. But the key step in phase two is building the temporal dynamics graph.
temporal dynamics graph?
This graph is UNAGI's way of being that cellular historian we talked about. It chronologically links the cell clusters across the different stages of a disease, say control, mild, intermediate, and advanced.
And how does it know which cluster turns into which one over time? It must be using some kind of, I don't know, sophisticated biological distance measure.
It is. It uses two key metrics to measure similarity across adjacent disease grades. First is KL divergence, which is a way to mathematically measure the difference between the distributions of two cell populations.
So how far apart they are, statistically.
Right. And second is DEG distance, which measures the difference in the ranking of their differentially expressed genes. By combining those two, UNAGI can map out these tracks that show the step-by-step changes as the disease gets worse.
Okay, so we have the map. Now we need to know what engine is driving the car along that map. That brings us to phase 3, the gene regulatory network, or GRN.
Precisely. To understand why a cell moved from state A to state B, UNAGI uses a specialized tool called IDREM, the Interactive Dynamic Regulatory Events Miner. Once that temporal graph is built, IDREM basically deduces the underlying GRN. It spotlights the key molecular drivers, like transcription factors, that are causing the changes along those tracks.
And here's the part that I found really fascinating, the part that makes UNAGI different, the feedback loop.
Yeah.
We find out which genes are driving the change, but how does UNAGI actually use that information to get smarter?
This iterative refinement is its unique strength. The disease-related genes and regulators that IDREM finds aren't just put in a report. They're immediately assigned higher weights and are emphasized in the loss function for the next round of VAE-GAN training.
So you can think of it like a biological filter.
It is. The model is forced to prioritize the critical disease genes over, say, generic housekeeping genes that are always on. And this constant feedback creates what we call disease-informed cell embeddings. The model gets smarter and more focused on the pathology with every single cycle.
That feels like the real difference between a generic AI and a biologically focused one, is learning where to put its attention.
It moves beyond just generic data analysis. It creates a system that has contextual biological awareness.
So let's do a quick recap of the blueprint before we jump into the case study. First, UNAGI uses a VAE-GAN and GCN architecture with a special ZLN distribution to handle that noisy single-cell data. Then it builds that temporal dynamics graph to map cell state transitions over time.
Right, using KL divergence and DEG distance.
And critically, it uses IDREM to find the drivers, the GRN, and then it feeds those findings back into its own training to get better and better. So what really stands out to you about how that iterative design changes the whole game for in silico modeling?
It's the fact that it doesn't just settle for good enough. It's actively challenging itself to become more disease-specific. It stops it from just modeling generic cell processes and forces it to model the actual pathology. And you see that power right away when we look at the case study on idiopathic pulmonary fibrosis.
IPF, it's a complex, horrible lung disease, irreversible scarring. It's exactly the kind of thing that needs this level of detail. But, I mean, you can't just take biopsies from a patient over and over for years. So how did they capture that progression?
This is where they used a really smart surrogate strategy. They used single nuclei RNA sequencing on 54 different lung region samples taken from 19 patients. And the key was how they categorized them. They classified the tissue samples into 4 grades of fibrosis control, mild, intermediate, and advanced based on alveolar surface density, which is a gold standard measure for scarring.
So by looking at different regions with different amounts of scarring, they could treat that as a proxy for time.
Exactly. It's a chronological proxy for the disease progressing.
That's a brilliant workaround. So what did UNAGI find in IPF? Did it, confirm what we already knew?
It absolutely confirmed the known hallmarks. For instance, UNAGI showed the proportion of fibroblasts, the cells that cause the scarring, goes way up.
Right.
It jumps from less than 15% in controls to over 40% in advanced stages. But more importantly, it didn't just count them. It identified 10 distinct progression tracks just within those stromal cells.
And inside those critical tracks, what regulators did it finger as the molecular criminals driving all that scarring?
So focusing on tracks like FibL4 and FibEV17, UNAGI found these pivotal regulators like CTCF, R8021, and SMC3. It also found a known bad actor in fibrosis, EP300.
Okay, so it's finding the known players and some new ones.
Exactly. And for pathways, and found the usual suspects collagen organization, PI3K, ACT, MT, or signaling. But it also highlighted less established pathways, specifically scalable signaling, which suggests completely new targets for therapy.
Now, how do we know this isn't all just a really fancy computational simulation that happens to look right? What's the proof that UNGGI is actually capturing biological reality?
This is where they did some exceptional cross-validation. The dynamic markers UNAGI identified genes like KEAL1A1, KEAL3A1, SERPINE1, which are central to scarring. They showed a highly significant overlap with independent proteomics analysis.
So protein-level data, not just RNA.
Exactly. Protein-level data. The chance of that happening randomly was tiny. The p-value was 9.354 times 10 to the minus 18.
Wow.
That level of corroboration tells you the model is capturing real physical changes happening in the tissue.
That is incredibly strong evidence. So once the model proved it understood the disease, the next logical step is prediction. Phase 4. In silico drug prediction, how did UNAGI go about searching for a digital cure?
It screened the CMAP database, that's the connectivity map, which has expression profiles from cells treated with thousands of different drugs.
Okay.
It was looking for drugs that could shift the gene expression signature of those diseased IPF cells back towards a healthier state. And it successfully found 2 known drugs.
Validating the knowns first.
Right. The FDA-approved drug nifedipine and another one called ifenprodil, an orphan drug for IPF.
So validating the knowns builds confidence. But what was the breakthrough, the big prediction that could actually be repositioned?
That was the prediction of nifedipine.
Nifedipine, the blood pressure medication.
The very one. A common established antihypertensive drug, a calcium channel blocker. UNAGI predicted it would have potent antifibrotic effects with an incredibly high score. I mean, an FBR of 1.152 times 10 to the minus 57. It blew the other drugs out of the water.
That is an astronomical score.
It is. It also flagged others like epicidin and salomalist.
So just to zoom out a bit, UNAGI uses this surrogate timeline, maps the disease, finds the drivers, and then digitally reverses the process, pointing straight at a cheap, established drug we already have. But of course, the critical moment is always the validation.
Always. Finding a candidate in silico is one thing. Validating it in a human-relevant model, that's where most discoveries fail. And they couldn't use animal models here. They're notoriously bad for IPF.
So they needed a human system. How did they test nifedipine in a way that gives us real confidence?
They used human precision-cut lung slices, or PCLS. This is a fantastic ex vivo model where you take thin sections of viable human lung tissue and keep them alive in a dish.
Wow, okay.
They induced fibrosis in these lung slices, and then they treated them with nifedipine.
And what happened? What was the outcome?
The experiments showed that nifedipine had antifibrotic properties analogous to nintedanib, which is the gold standard. But crucially, the in silico prediction results lined up almost perfectly with the real PCLS treatment outcomes.
Oh, perfect.
For the top 100 differentially expressed genes, the prediction showed an R-squared score of around 0.889.
That is a massive correlation. That R-squared value is basically saying UNAGI's digital prediction almost perfectly tracked the actual biological reality.
It's the proof that the digital manipulation inside the computer accurately predicted the physical effect in human tissue.
Amazing. Now, beyond IPF, did they test its versatility? Could it work on a completely different disease?
They did. They applied it to COVID-19. They analyzed almost 250,000 peripheral blood mononuclear cells, PDMCs, across 4 severity stages of the disease.
Then what did it pull out of that? I mean, COVID is all about immune cells, not structural lung cells.
It successfully identified key regulatory networks in COVID progression. For instance, the RHO-GT paces activate NADPH oxidases pathway, which is very relevant to severe inflammation. It also predicted drug candidates like aloxastatin and adenosine, so it clearly shows it can adapt to totally different biological systems and cell types.
And finally, how did this super specialized, disease-informed model stack up against the more general tools out there like SCVI or even the new foundation models?
It consistently outperformed them. It got higher scores on metrics that measure the quality of cell type embeddings. But most importantly, for in silico drug screening accuracy, UNAGI had a superior AROC of 0.945.
And the researchers think that's because of the feedback loop.
They attribute that superiority directly to the iterative, disease-informed feedback loop we talked about. By constantly refining its focus, it just generates more relevant insights.
We should probably mention any limitations.
Yes, there are two important ones. First, its drug screening is limited by what's in the CMAPE database, which is not, a complete library of all drugs. And second, while it's great at decoding disease progression, it wasn't specifically designed to classify individual patients as, say, progressors versus non-progressors.
Those are important caveats. Still, UNAGI seems like a major step forward. It's integrating AI with deep cell biology, moving us from generic models to these disease-informed systems that can decode progression.
Wow.
And this is the big one, accelerate drug repositioning.
It totally shifts the question. We go from what if we try this drug to what drug signature will mathematically reverse this disease.
Exactly. So UNGI's ability to model and manipulate these cell dynamics was proven by predicting drug effects on disease cells in IPF. But here's a final thought for you to consider. If we can predict therapeutic outcomes and push disease cells toward health with this level of accuracy, what could a tool like UNAGI teach us about controlling healthy cellular programs? Like manipulating cell fates during natural processes, things like embryogenesis or organogenesis. That really feels like the next frontier of cellular engineering, and it's a question for you to mull over.